Hello, everyone. Namaste. Good morning, good afternoon, and good evening. I hope you are doing all great. Well, it's late evening here in Nepal, and we are here back again with brand new episode of webinar on Geo-Science for Sustainable Development. This is Suraj Gautam, a civil engineer and a disaster risk reduction and management professional from Institute of Himalayan Risk Reduction. First of all, I'd like to welcome you all in our Himalayan webinar series. Institute of Himalayan Risk Reduction is an organization established with a vision to prepare risk-informed societies for disaster resilience and sustainable development. So to contribute onto this, IHRR has been regularly organizing Himalayan webinar series on geoscience for sustainable development and disaster risk reduction and management. So far, we have successfully organized 6 episodes of webinar on geoscience for sustainable development. And yes, today it's the turn for the seventh one. Today we'll be having an interesting topic on understanding of sediment provenance and diagenesis using stable isotopes in phosphate, which will be presented by Dr. Dev Jaisi. On behalf of the organizing team, we are really grateful to our presenter who will be sharing us with his insights and healthy experiences. So it is indeed an honor to welcome our presenter, Dr. Dev Jaisi, who is an associate professor at Department of Plant and Social Sciences, University of Delaware, Newark, USA. To make this session more interactive and lively, we have Assistant Professor Dr. Basanta Adhikari from Institute of Engineering, Tribhuvan University as our session moderator. And Dr. Adhikari is also the visiting scientist at Institute of Himalayan Risk Reduction. We would like to we'd like to welcome all of our participants and regular attendees who are joining us through our social media live streaming and also through this Zoom platform. This webinar event has also been featured in the website of Relief Wave, which is under OCHA services and is the largest humanitarian information portal in the world. Talking about the webinar registration, till date, we have 175 registrations from different parts of the globe. So to address this request, we are also going live on our social media page of Institute of Himalayan Risk Reduction. We'll be receiving the queries and feedbacks from our social media live streaming and as well as from our Zoom chat box. And yes, we have been organizing this webinar series on geoscience for sustainable development on a regular basis. We believe this webinar series has been supporting our early career scientists, DRR professionals, academicians, and to all the participants. And yes, it's just a beginning and we'll have a long way ahead. So before starting our session for today, let me share some brief introduction about Institute of Himalayan Risk Reduction. Well, Institute of Himalayan Risk Reduction is a research institute established with an objective to assess and understand the risks to contribute for the sustainability. IHRR works under the 4 thematic areas that are DRRRM consulting, research and development, disaster academy, youth in disaster risk reduction and management. So our team constitutes of professionals with expertise and experiences in the field of DRR in understanding the risks, conducting academic research and field level implementations in Nepal and across the globe. So far, we have accomplished a number of projects that includes the recent one where we had a collaborative approach with a number of organizations, and we contributed in the development of national platform for the information dissemination of COVID-19. So the app named Hamro Swasthya was developed, and it was also endorsed by the Ministry of Health and Population. So it is also it is being widely used during this period. And besides, we have also collaborated with the local governments, and we are working with them closely. And we, using this lockdown period, we carried out UAV surveying in Hetauda sub-metropolitan city. Uh, and we have also published our commitment in SENDAI voluntary commitment. And apart from this, we have been regularly organizing this webinar series, Himalayan webinar series. And today we are here on our yet another episode on geoscience for sustainable development. So before I would like to start our session, I would like to share some house rules for this webinar. The audios and videos of the participants will be switched off. The overall session of the webinar will be recorded. Only for any queries, the participants can write up in the Zoom chat box, as well as they can also write in the Facebook live streaming. The waiting room has been enabled, and only registered users with their original identity will be allowed to enter the room. Also, I would like to request our participants to please rename and keep their name as it was mentioned in the registered link. Also, I would like to request our participants to strictly avoid using the annotations. Annotating during the presentation will be taken offensive, and the participant will be removed from the webinar. The feedback links will be provided during the session, and we'll be providing certificates, participation certificates. We'll be sharing the certificates in our website soon. So without any further delay, I would like to move on, and I'd like to, may I request Dr. Basanta Raj Adhikari to proceed further with his moderation. Thank you for now.
Thank you, Suraj. Am I audible now?
Yes, you are audible.
Thank you, Executive Director, Engineer Suraj Gautam, for providing me opportunity to moderate this session, to our esteemed professor, our senior geoscientist. Actually, he's my teacher. He taught me during my undergrad at Tribhuvan University, Trichandra campus in Kathmandu. And today we have requested him to present his research to our young scientists and established scientists to understand some of his research on this phosphates and others. So today, Dr. Dev Prasad Jaisi, who is Associate Professor, Department of Plant and Soil Science, University of Delaware, Newark, USA. Dr. Jaisi received BS and MS degree from Tribhuvan University, Nepal, a second master's degree, MS degree from SN Institute of Technology, Thailand, and PhD from Miami University, Ohio. He was a Batman Scholar at Yale University before joining the University of Delaware. He currently holds visiting professorship at California Institute of Technology and Huazhong, a University, China, and is senior visiting fellow at Xiamen University, China. He is an associate editor in clays and clay minerals, +1 soil system and pedosphere, and editorial board member in ACS Earth and Space Chemistry and Soil Methods. Dr. Jaisi has published more than 73 peer-reviewed papers and book chapters with a Google Earth Index of 30, which is very, very impressive. His research outputs are included in state and federal advisory board reports on water quality, including the annual science report, draft to the White House, and in +20 local plus regional media news. In the past 9 years, his research generated 9 million federal grants, US dollar, and awards. He is a co-founder and co-director of Environmental Isotope System, a central facility at UD. He serves in various regional, national, and institutional professional organizations and advisory councils. He received National Science Foundation's Career Award and EPSCOR Fellowship, ACS Early Investigator Award, and ORAO Innovation Award. And Dr. Jaisi is famous not only in the United States, but around the globe, and he's more famous in Nepal and this region for his continuous research work and contributing science to the society. So today he's going to present on understanding of sediment provenance and diagenesis using stable isotopes in phosphate. So Professor Jaisi, the virtual floor is yours. Please proceed.
Thank you so much. I'm very honored to be a presenter of this series, and I really appreciate the effort from the IHRR for youth, largely motivating through connecting the national and international expert. That was a very humbling introduction, Dr. Adhikari. I really appreciate that. Without doing any delay, let's start my talk. Okay, so today I'm going to talk on understanding the sediment provenance and diagenesis using stable isotopes in phosphate. So I'll give a general outline of my talk today. First, I'm gonna put some very generic info on phosphorus and what research area we work in my lab. Second, I will introduce the phosphorus cycling, including the major research method my lab works on. Third, field site and sample collection and processing. And then I'll move on results and discussion and wrap up with conclusion and bring up with some further reading or connection that some of the young generation in Nepal would be interested to doing similar thing in my lab or around the world. All right, so phosphorus.
If you have a problem, okay, fine, yeah, go on.
It's like it's going on, okay. So phosphorus is a mystical and magical element. If you want to go back to periodic table, it's the 12th element to be discovered. At one point, there was a element of Ross, like a gold Ross in 19th century, right? So every big scientists were going after to find an element. And a person in the right, which is Henning Brand, at 1669 did some very uncommon experiment. He thought, well, I'm gonna get gold out of my urine. So he was processing his urine. He ended up getting an element, a bright yellow, which is phosphorus. So it is a very odd way of identification and coming into the world of science. As you all know, phosphorus is an essential element of life. Every piece of all living being has phosphorus. It's built around this planet for about 4 billion years. Just not the structure, but energy storage, expression of heredity, cellular energy, and functions are all centered around phosphorus. So if you just Google out why phosphorus in a living being, there are about 13 or 14 Science and Nature paper. It has been a element coined differently over time. Early in the 7th century, it was considered as a philosopher's stone because a philosopher found out, and then was used for medical purpose for longer time. Then it gradually entered into other applications like flammable property, using phosphorus as a nutrient for crop, and then also used as a war element. So elemental phosphorus is a very vigorous reaction, so used as a white phosphorus as late as 2005 war in Middle East. Now it's a eutrophication problem, and in future we're looking at phosphorus scarcity. Let me look at the two sides of phosphorus, which I always enjoy talking to my students. The phosphorus has two sides. If you go to periodic table, I do not believe you'll find an element which has a two extreme end. Look at the left. If phosphorus is a partial ester, it's always play essential elements for life and plays a vital role. For example, I mentioned before, every part of life are built on phosphorus, center on phosphorus. If you are a historical geology, paleo Earth, then you know that the evolution was paused significantly because of not having enough phosphorus in the planet. And now, maybe from last about 100 years, green revolution, we have been enjoying a variety of food in an easy and a very clean and pristine way of growing them. On the right is a neutral ester. So if the phosphorus remains as a neutral ester, it's a biocidal activity. That means it kills anything and everything. I mentioned before is a warfare agent. For example, choline esterase inhibitor. That's the major role that phosphorus plays. If you read the BBC News, just until yesterday, there was a Russian opposition person who was fed with a compound which is choline esterase that just disturbed the cholinerase system in our body. So person paralyzed right away and dies in a matter of minutes, depending on the dose. Many insecticide, fungicide, nematicides, rodenticides are made from phosphorus. And the last is my interesting part, drugs like sulfobuvir for hepatitis, brincidofovir for Ebola, and remdesivir, COVID-19 drugs are all based on phosphate. So just a unrelated note here, I'm working on this remdesivir drug for coronavirus treatment as well. So my team are working hard to get the pathway of this drug transport inside the cell. So in essence, as you see down here, two pictures that both life and death of all living beings in this planet revolving around phosphorus. So think of any other element in this planet in the periodic table that does those two extreme. It's only phosphorus. So I hope I energize you enough that you can be interested or want to be interested working on phosphorus. Let me highlight a couple of work that my lab at UD focuses on. So bigger umbrella is a phosphorus biogeochemistry. Below you see our interest in fundamental processes of reactions. For example, bacterial respiration, minerals precipitation, dissolution, interface reactions. In all cases, we have a single question. Are there any reaction-specific, pathway-specific, process-specific isotope effect? The reason we seek that question is that if we know that effect, then we can interpret either present or past environment very confidently. Basic knowledge we gain from this fundamental science, we apply in various ways. The organic aspect of research is formation and degradation of organic compound. We use both enzymatic and abiotic processes. It includes series of organic phosphorus listed here, and as well as the drug compound, which are organic phosphorus compounds as well. The major part is the application of, we learn from these two areas of fundamental science to identify sources and cycling of phosphorus at the ecosystem level. So these are the site, Chesapeake Bay, Mississippi Delta, and Cape Cod in the US, but I have a series of collaborative projects all around the world, from Chile to Europe to China, and so on, and that list increases over time. Let me move you into phosphorus cycling. So if you look at the phosphorus cycling, it's good to divide into terrestrial part and marine part. So terrestrial, the phosphorus primarily come from weathering of the rocks. So we mine from what comes of the earth processes into some deposits, economic deposits, and apply to plants. So weathering and erosion transport phosphorus from land to ocean or any other water body. In the water body, it could be used in the upper part of the water column and then gradually settle down and then goes to the sediment. So today, I'm gonna focus on the sediment processes of phosphorus cycling with some detail. So what I'm showing you is some of the research that I did when I was still a postdoc researcher at Yale. This is Hawaii. As you know, Hawaii has the biggest island. It's a big island. The right, lower right corner, and it has a small seamount, which is under the water. And this is a research vessel we used to go to have some study on phosphorus cycling. This is a cruise, as you can see me, the second from right in this bright sunlight in Hawaii. This is a general series of a picture on the showing processes. Our university has a research vessel. It's called RB SARP on the top left. And the right, you see my postdoc and graduate student using a smaller boat, go into bigger boat. And the right, you see a core sampling. This is a multi-core sampler. We release or drop into water body that goes all the way at the bottom and then insert into the sediment. And then sediment is packed well in that column. And below you see how solid the sediment cores come in. We slice that core into different depth. And as you know, it recurs the sediment of past history, right? The topmost being the most recent, with the deeper being older and older, which using various geochronology, we can identify how old the sediment so that we can reconstruct the past history. And once those sediments are sliced, it requires a series of processes for processing until eventually it precipitates into silver phosphate for isotope analysis. So I'll dive you a little bit more on how we process this sediment until we have a final analyte. For example, here you're seeing a pieces of sediment, a dry sediment. On the right, if you do a SEM image analysis, you see a clay mineral, some biological shell. And if you do a EDS, that's an elemental composition of that particular position. For example, here you would see A being A site, largely silica and other elements, oxygen, sulfur, potassium, calcium, and so on. Among a spatial location, the composition of those elements could vary depending on what composition of mineral we have. In the A, you see there's a very small peak of phosphorus you barely see, and that's the elements of my research interest. And you also notice that the amount of phosphorus is so low, way lower than 1%, much lower than 1%. So it requires lots of sediment or water to process for analysis. So let's think this way, that the soil or sediment is taken in and only phosphorus is extracted. We can do some sequential extraction, that's a step-by-step extraction, and then quantify them. For example, here you see the list. One phosphorus pool is for water phosphorus or water-soluble phosphorus. Second is loosely sorbed phosphorus. Third is occluded, co-precipitated with iron and aluminum oxide phosphorus, authigenic phosphorus, detrital, and residual. So for the geological or paleo-environmental implications, the fourth and fifth, authigenic and detrital, these are the two pool I'll focus, so please keep that in mind. The beauty is that as the arrow pointing down, the phosphorus in the lower numbers are less and less bioavailable. That means they preserve the paleo environment or paleo signature of life, so we can extract paleo environment for any sediment of interest. So as you know, authigenic phosphorus is phosphorus that is precipitate. We're talking about mineral here, not the phosphorus element is always there. Phosphorus precipitate as an authigenic mineral in situ precipitation in the sediment. And detrital phosphate is, those are the mineral, they are precipitate out of the basin. For example, could be a volcanic environment in the land, could be just a generic upliftment of the basin plateau, and so on. So let's keep in mind on these two phosphorus pool, authigenic and detrital, for the next stage of paleo-environmental reconstruction. So I'm showing you a pretty busy procedure here, but I want to wrap you up. Just look at the right side. If you have a sediment, you have a series of chemical reagent you can use to extract phosphorus pool, I mentioned before, exchangeable, and lowest down to are calcium P, authigenic phosphorus. Calcium P is a same mineral form, it's just the authigenic and non-authigenic phosphorus as an apotype. So those two are the elements, those are the minerals of our interest. I will dive deeper into. And so what I just said before here was the phosphorus pool. If you have a sediment, you extract into those different pools. Once you extract, the solution has other component beside phosphorus. So for isotope analysis, all these contaminant except phosphorus have to be removed. And as you can see, every arrow here is the reaction. So it requires a series of processing until a final pure phosphate mineral is formed for isotope analysis. So process-wise, it's a little time-consuming, and many times I take a light approach to encourage the students. Say, if you do it correctly, you'll be very happy. Your partner will be very happy because you learn lots of patience of doing science correctly. This is the isotope facility in my lab. So what we do is TCEA, it's a thermochemical elemental analysis. So what it does is pyrolysis. That means burning of material in the absence of oxygen. So you have a phosphate mineral, PO4, which is burned down, as you can see here. And this PO4 releases oxygen, and that oxygen goes as a CO gas, and then isotope of that CO gas is measured, and oxygen isotope eventually calculated. So let me be very brief. From the sediment, different phosphorus pools are extracted. Those pools are further processed to make a pure phosphate, and the oxygen in phosphate is analyzed. Not the phosphorus in phosphate, but oxygen in phosphate is analyzed to track phosphorus, because phosphorus is almost always present as PO, largely PO4. So analyzing O tells the story of P. Okay, the reason we cannot do for P is P has only one element, only one stable isotopes, 31. So we cannot use a stable isotope approach. However, that's a benefit of using other method, for example, NMR. So P31 is a very NMR active element. We can identify organic species and information using PNMR. So let me bring you to the field site sediment water sampling. So I'm going to bring you down all the way to South America. Let's take an imaginary flight there. You see a country, Peru, off of the Peru here, which is a very active upwelling zone where active phosphogenesis is happening. What does that mean? Is that the formation of sedimentary phosphorus are active there. So we can look at how phosphorus actually deposit in current time so that we can go back at the past history. There are a series of sites on this Peru margin, and I'm gonna lay them here at site 12, 27, 28, and 29. One thing is easy to remember, it's a 27 being the deepest in terms of water column, which is about 500-meter deep, and 1229 is the shallow, so about 150 feet. Majority of work are focused on that middle site, which is 1228. So let's look at the sediment property of that site in terms of phosphorus. So this is a field drilling activity. Those who are new in this world, you have a seagoing research vessel up here, and it has a drilling rig that goes through the water and toss to the sediment water interface and drill down into the sediment and collects the sediment. Bottom, you see a unmanned rover that you can deploy to look at the sediment surfaces. On the right is the lowering of those sediment coating device. On the right top, you see the scientists are bringing those sediment core inside the lab. As you can see, they have a face mask and oxygen tank on them. You might have thought maybe these sediments are anoxic. So the entire room there is anoxic to maintain the sediment property. So it's a delicate process and lots of care needed for scientists. And below, you see they have a fun component, humor component as well. When they get time, they always enjoy taking picture and having cracking jokes. So let's look at some of the results gotten from those sediment core of Peru. I'm only showing the sediment pool. On the left, I'm not sure how visual for you, these are late Miocene to Pleistocene sediment, goes back to about 40 million years. So the lowest sediment, which about 130, 40 centimeter depth, goes back to about 40 million year old, and the top at 0 is the most recent. As you can see, the phosphorus concentration in micromole per gram. So mole, millimole, micromole, it's a very small quantity, but you see a particular pattern of phosphorus concentration. At the top, very shallow, you have those iron oxide bound phosphorus, which become less and less at the bottom. Again, organic phosphorus very high at the shallow, become less and less. What you see is that authigenic phosphorus increases significantly at the depth 100 meter and below. So let's look at carefully what causes, what are the sediment processes that we can extract from measuring isotope of this phosphorus pool. This is the isotope value of those 3 sites, and at the center, 1228 site, there is a zoomed in version as well at the very shallow depth, 0 to 6 meters. As you can see, the isotope value is quite variable from almost 23 to 24. And over the spectrum, you have isotope that expand from almost 20 to 24 per ml. And let's look at what causes that variation, or what's the likely interpretation of those variation of isotope. You see a solid line. That solid line refers to equilibrium isotope value. What does that mean? If phosphorus is fully bioavailable, that means it's a fully cycled bioorganism that's living in the pore water or attached to the sediment, that's where the isotope you expect. So any isotope that is within or nearby to that line are equilibrium or fully biologically cycled phosphorus. The beauty of sea cruise is that as lots of expertise, they analyze different aspects of science. Here, I'm just showing the temperature. So temperature of a sediment could be variable over time. The paleo temperature versus the current temperature are different. Here you are seeing the ancient temperature in datas, different plots from a smaller triangle to bigger triangles, and some circles as well. The current temperature, all plus and a solid line. So clearly there are differences in past and current temperature of those sediment profile. And that's a benefit for us for paleo environment reconstruction. So now I'm going to show you all a component of this slide first. As you can see, we can group those phosphate into three larger components. One, that's oval, as you see, which is far away from equilibrium, whether present or past. And what you see with that solid line I mentioned is equilibrium at current temperature in the sediment, and the gray zone is a paleo temperature equilibration. So these three group of phosphorus mineral are formed under those three different circumstances. So that means when the sediment were depositing, there was an active phosphogenesis. That means phosphorus minerals were formed at that time and continue to build on those sediment. And the solid line, the data that are close to solid line and fall within solid line are the current temperature or current time. So phosphogenesis process still exists in pore water in the sediment. Even the magnitude of that could be much lower. What you see on this big oval is that couple of data points which are very off of equilibrium. The only possibility of this isotope is that they are off of the equilibrium because of extensive hydrolysis of organic phosphorus. So what does that mean is the water column above it is a very fertile, at a lots of microbial activity. So they grow and die, and then those dead body or molecules of organic phosphorus in the body gradually settle at the sediment and then break down to form phosphate. So we have a three generation or three larger classes of phosphorus, and that aid on identifying phosphogenesis in the sediment. So just for very short, I'm gonna pitch in here that the formation of apatite in a water column sometime is possible. It's a busy slide. I'm not gonna go detail, but I'm gonna show the left image where you can see a bright P written, the phosphorus element, the bright, the little bit elongated, obvious looking figure is for phosphorus. So these are precipitated in the water column in a very rich environment. Now, what I'm gonna do is a reconstruction of phosphorus processes in the water column and very bottom in the sediment using the data obtained from NMR analysis of composition. So I'm listing here series of organic phosphorus in organic debris. Let's say phytoplankton, diatoms, all kinds of organisms are in the surface, active, and once they die, either they are chewed up right there or they settle down. Let's take the component that settles down, and we can look at the composition of organic phosphorus at the top, number one up here, and continuously at the bottom. And what we see based on the composition of phosphorus at the top water and the bottom water, we can identify how the phosphorus cycling is happening and phosphate is precipitated in the sediment. A little bit of detail on the isotope on the left. I would not go for now unless a detail is needed and someone raises question down. But in a water column process on the right, you see the phosphorus from dead organism at the top water, photic water with its light, the lots of living being, they gradually settle down and then hydrolytes break down during the process, and some part goes to the sediment. In the sediment process, the recycling happen during early time and during diagenesis and precipitate as different generation of apatite mineral. So the beauty is that looking at isotope value of this apatite mineral, we will be able to identify the environment when those minerals were precipitated. Let's say if your interest is to go very deep into Earth history or want to go to the out of the Earth, let's say Mars, and ask a question, well, other life are there or there's a biological cycling. Looking life is not finding a living being, but finding a signature of living being. For example, traces, molecules that are pertain or produced from living being in the past. So isotope is a proxy for that. Now let's move to the detrital. My apology, the title should be detrital phosphorus. Apology again. So I'm showing the detrital phosphorus of these three sites, and you see very similar way detrital phosphorus fall into two categories. One I showed the horizontal hatch, and the other is a vertical hatch. And if you go back to the history of this area, as you can see clearly, majority of river just flowed away from Peru. So they start in the top part of Peru equator and go to imagine all the way released to the right side of the figure. Peru has a very small river or rivulet there, and that erodes the sediment from those high latitude, brings down and deposit on the ocean. So the beauty of detrital phosphate is that because they preserve the isotope value, those two regions are one from detrital apatite in igneous rock. So what happens is that you have igneous rock up there, those igneous rock have apatite, those apatite are transported as apatite grain and then go to the sediment and deposit there and remain as a detrital grain. So these are the one classes, the light isotope value. The second vertical has are phosphate. These are actually precipitate on the land. So in the some environment where meteoric water interacts and meteoric water help extend some level of isotope, and that range is precipitated. For the second range, there are some other mechanism that might also result similar range of phosphorus, which I will not discuss at this moment. But most likely these are ex situ, that means land precipitate apatite that transported to the machine of deposition. So let me summarize that isotope is a very useful tool that we can identify multiple origin, both intra and extra basinal, quite useful. Isotope of phosphate is a stable proxy and may not be spaced there, alter in geological time scale. And the last, which is more relevant to most of the modern environment, is that the phosphorus mineralization is a dominant pathway, which is perhaps a new paradigm of phosphorus cycling in coastal environment, eutrophic phosphorus-rich environment, and that has gotten quite a bit of interest from our output to the national level news. So let's go a little bit beyond where this isotope use be done. I'm just showing we can go as far as Archean Ocean. I'm not gonna go detail here. This is a Nature paper from my advisor, Ruth Blake. at Yale. She was able to identify the Paleo-ocean, the Archean ocean temperature using phosphate oxygen isotope in banded iron ore formation. That's in South Africa. As you can see, nice bands on the top right side picture. Those banded iron formation are formed in Archean time. The beauty is that this is a very tectonically inactive region part of the world, so that there is not much deformation going on after sedimentation, so that those isotope value remain the same over that long time, and was able to identify the paleo environment of the ocean was much milder, about 25 to 30 or even 35 degree, than all other tool says about 45 or so degree. Let me wrap up here with area of exploration, Nepal, Himalaya, or around. So geocentric area, if your interest is sedimentation, formation of stromatolite. You see a stromatolite picture when I was doing my thesis in Nepal. So I took lots of those pictures, and I have a vivid memory of stromatolite and perhaps identifying the environment of deposition at that time. Other could be sources of sediments in Siwalik, any formation and post-depositional environment, for example, calcite, dolomite, lots of resources, a huge isotope laboratory. Those could be used for those analysis. Right is the environmental centric area, which largely my lab focuses on phosphorus chemistry and processes at diagenesis scale. I showed you on my third slide, phosphorus cycling in water and impacting in water quality, and various processes like mineral formation, dissolution, and interfacial processes at micro and nano scale. So if any of this area interests you, what I would like you is to reach me in my email bottom here in the right, jaisi@udel.edu. Just keep in mind, sometimes people say JAISHI, and the H has to be gone. So just jaisi@udel.edu. I'll be responding and exploring area that you can work on. On the left, I'm showing a Blake lab where I was a postdoc, because I presented most of my work done during that time. So credit goes to my advisor at the center, Ruth Blake. In the right, these are my student, a postdoc in my lab. So you can see all are energetic and they work very hard. They are very intelligent. I'm really proud of all of them. And my funding agency from NSF, USDA, and NASA. With that being said, I'd like to thank you everyone, listen so passionately, and I'll be happy to take any question further.
Thank you, Professor Jaisi, for your wonderful presentation and lots of examples from North America as well as South America, which are really, encouraging for us to understand this biogeochemistry and the cycles and how these phosphates are really important for human being, not only human being, but also to reconstruct the paleo history, what you have rightly mentioned about this temperature, calculation or, understanding of temperature of Archean Sea based on these studies. That is really, really encouraging and wonderful. And I'm sure that there will be lots of questions both in Zoom chat box and Facebook, but I'll take a question first from Facebook from Soni Mulmi Pradhananga. So asking, Namaste Debji, it is great listening to your talk. Can you elaborate a bit on instrumentation that you used for element analysis?
Sure. Thanks for the question. So we have ICP, it's inductively coupled a plasma mass spec that measures a majority of elements in periodic table. So that's for the element analysis. If you want to know the concentration or amount of elements of interest. For the isotope, it's called isotope mass spectrometry. It measures the isotope of those elements, which I share the picture. So both equipment, my lab has, and my lab members are very well trained on that. So if you see use of those elements analysis in your project, please contact me.
Thank you, sir. Another question again from Sony. So question is, was your carbon and oxygen isotope analysis able to distinguish sediments derived from wind-driven upwelling and primary productivity? So I'm just curious if you were able to come up with the sedimentation rate to suggest source and formations.
Very interesting question. It has a several component. So for the sedimentation rate, there are other more reliable proxy, perhaps a radiometric method, which I also have a lab, bigger lab for those. Gamma counting takes a long time, but you don't need to sit there, could be measured easily. In terms of sources like an aeolian deposit, so let's take this way. If we have a 2 or 3 vehicle coming at one point, how different those vehicles are before they come in there. So that determines whether we can distinguish their origin or their sources, right? So many of the aeolian sources have distinct isotope, and many times the other element that gives a signature of particular sources. So one of my students does this vector modeling. So she uses 46 number of elements and use a matrix-based method. The beauty of matrix is that you can have up to 96 parameters, variable parameter, dependent parameter, have a one independent parameter, and test likelihood of particular sources. Perhaps I took a broad brush here to narrow down your question. It is likely that the dust or aeolian sources of phosphorus could be distinguished from isotope, but you need a little bit of other method if your question is on sedimentation rate.
Yes, thank you very much. And some questions from the Zoom chat box. If you can't really type your question, and if you are from mobile, and if you want to ask by in voice, you can just raise your hand and we'll entertain 2 or 3 questions. So another question from Anup Neupane. Thank you, sir, for your nice presentation. Which technique do you use to analyze the age of sediments?
That's a good question. So the analysis or age for the sediment is not my expertise. So these are done from other researchers. They use radiometric technique to identify the age of the sediment.
Okay, thank you. So question from Purna Kumar Khatri is that what is the mean delta value of organic phosphate? How did you prepare organic phosphate sample for the analysis?
That's a very interesting question. What's the name again? Who raised this question?
Purna Kumar Khatri.
Okay, Purna Kumar, you have a gold medal there, okay? That's a very interesting question, and we have a huge energy to work on that. So let me give you a theoretical aspect here. So let's say we take food, has a phosphorus, goes inside our body, and those phosphorus compounds are broken down into element. Again, they become as a phosphate all time. We don't break into P and O, remains as a PO4. And that time we have an enzyme involved. That enzyme imprint the new isotope value. So any organic phosphorus that goes up throughout my body or organism have a distinct isotope fingerprint, which is caused by enzyme in that body. Because enzyme is the only factor that cause two-way isotope exchange and then imprints that isotope signal. That means unless that phosphate is utilized by any other organism, the isotope remains the same. So that's the beauty of using apatite, identify what happened before it formed apatite, right? So we have developed a new method to identify the isotope value of this organic phosphorus compound. But all classic methods are only for inorganic phosphate. So PO4 in mineral, not in an organic moiety. So if you have an additional question on organic phosphorus, their isotope fingerprint and how they are derived or what they can tell, I showed you my email, which you can Google as well. Please write me email. I'll be happy to talk to you further.
Thank you. Another question from Dibbe Nan Dubey. Thank you so much, sir, for your excellent presentation. So tell me the number of isotopes of the phosphate, which one is most stable?
So again, we're talking about stable and radioactive isotopes, right? So phosphorus has only one stable isotope, which is P31. Phosphate has oxygen, so that oxygen has 16, 17, 18. Of course, there are other stable isotopes, which is unusually low. I don't think we'll be able to measure in the next 50, 60 years, unless there's a huge revolution on the technology, right? So among these 16, 17, and 18, 16 and 18 are stable isotopes. These are mass dependent. So that means their property are driven by mass dependent earthly processes. Mass 17 is mass independent fractionation. So that's more of an extraterrestrial process. So if your interest is to look at what phosphate, part of phosphate is earth processes versus extraterrestrial process, you have a mass 17. You heard that I'm a visiting professor at Caltech. So Caltech is a front line research in extraterrestrial life, right? Planetary science. So I'm exploring that world as well. So yeah, it is a further question. Please drop me an email.
Thank you, Professor Jaisi. I think I will invite some of our senior professors from Tribhuvan University. They might have some remarks on your presentation. So Ananta sir, are you there? Suraj, can you just unmute Professor Ananta Gujral?
Yeah, sure. He's already unmuted.
Anand sir, are you there? No, he's not unmuted.
He has to do.
Anand sir, are you there? Okay. In the meantime, I don't know something. Anand sir? I think he has some problem. So I would like to request Professor Dr. Kumnaran Paurel sir to give some remarks. Suraj, can you unmute Professor Paurel?
Yes, he's already unmuted.
Come sir, are you there? Sir, can you unmute by yourself? I think he's also having some problem. So in the meantime, I would like to ask Professor Dr. Santaman Raiser. Raiser, are you there?
Yes.
Yes.
Yeah.
Please give some remarks.
Hello. Yes.
Yes.
Yeah. Namaste, everybody. I'd like to thank you very much, Professor Jaisi, for your nice presentation. And it was a really interesting presentation. I was so happy to hear your presentation, your research, your ongoing research. Of course, I'm not in this field, but I learned something from your presentation. Thank you so much. And I have some question because it's really, I've never done research on isotope, oxygen isotope, or in your field, but I have some interest if you would like to conduct some research in the Himalaya. For example, there are some phosphate deposits in the Gondwana sediments, and if you plan to see the phosphorus content either in the Gondwana sediments or in the Indian Ocean, there could be really big contribution for the Himalayan scientists. And of course, you have doing very nice nice research in the mostly in the South American and American area in the ocean. I have some question, Devji, if you would like to. Uh, I don't know. I am very interested to know the content of distribution of phosphorus sediments. If you see the content of phosphorus between the terrestrial zone and ocean zone, because you are doing the research, I saw in the South American Ocean, and you have collected some of the sample close to the in the self zone. If you go further from the continental, part, and did you find any difference in the content, phosphate content, and how do you link between the source or provenance and the ocean sediments? Could you tell me, please? I feel it's very interesting. Yeah.
Sure. Thanks for the question, Raisa. So first question is doing some phosphorus content analysis on Gondwana and Himalaya. I'll be very happy to, if there's any student who wants to join my lab for those questions, I encourage them to contact me. So looking for those way to connect to the Himalaya and broader aspect of research in Nepal. So, I'll wait to hear anyone with interest. Regarding the phosphorus concentration distribution, phosphorus is always a ppm label, 0.001% or low concentration in general soil and sediment, except pedogenesis that allows the phosphogenesis, that means phosphate is precipitated. The reason, again, this research is just a small part of a big part of my research strength and area we're working on, is that area has an active phosphogenesis. It's an active upwelling. So in some part of the ocean, the bottom water comes all the way up. So that vertical movement, upward movement of bottom water. And those kind of environments are considered conducive for precipitation of phosphates. That's the region that the study was set aside, was chosen, and that has a very high phosphorus concentration compared to sediment. If we go to the deeper water towards the main ocean, the concentration decreases significantly. And if you go all the way to mid-oceanic rise, where the tectonic boundaries are and where the hydrothermal activities are, that particular part also has high phosphorus. So the hydrothermal vent and processes somehow gets quite a high phosphorus concentration, but middle part of the kind of a dead ocean where there's not much tectonic activity has very low phosphorus concentration on ppm level, parts per million level concentration.
Thank you very much. Thank you so much.
You're welcome, sir.
Thank you, Professor Rai and Professor Jaisi. So I don't see there's some question in Facebook. So let me see in Zoom. Most of the comments, not questions. So yeah, I have some observation, Professor Jaisi, because the best part of the phosphate and others which you rightly mentioned about this biogeochemistry or biogeoscience or whatever you can relate on this understanding of broader aspect. Most of our participants here in Zoom and Facebook are from this Himalayan region, and which you have rightly mentioned about this future possibilities to do research in that region. And thank you very much for your warm welcome to our new graduate student who really want to join to your lab to understanding, because you have rightly mentioned about this stromatolite which we can find in Thading dolomite, and then other Gondwana sequence which Professor Rai has rightly mentioned. It's always good to see such extensive research experiences to share with us, and we are very, very lucky and we are very honored to have you here, sir. So before I hand over to Suraj Gautam, do you want to say any final words, Professor Jaisi?
Sure. So my reference is that if any component of the research that I presented or I pointed towards possibility, please reach me. Or if you see anyone that might be interested, please share. So my lab always have lots of assistance, funding opportunity. Luckily, I've never been a funding issue. I always have a couple of million to spend. So that is never a problem. What I really ask you is that also watch whether you have a right background or you are very ready to jump in the fire and then turn the fire into a gold, right? Luckily my group has very, very motivated students, and they're gonna really help you to fire up, but please judge that. And if there's any question, you can always reach me. You can always reach one of my students. They're gonna say how my lab look like. And if any of you wants to join my lab, I would take the opportunity, because my part of doing something science in Nepal has been falling behind, which I want to raise up. Thank you.
Yes, thank you, Professor Jaisi. And it's, we always welcome you to do a research in Nepal, Himalaya, to invite our, young graduates, which is your route as well. And we have to train our new graduates in this field in different arena. And thanks for your welcome and very kind message to our young graduates and researchers back here. So thank you very much, Suraj, for providing me this opportunity to moderate my professor and esteemed colleague in Geo-Science, Professor Dr. Dev-Prasad Jaisi. So the floor is yours. Please, Suraj, continue.
Thank you so much, Dr. Basanta Adhikari, for the excellent moderation, as always. And we would like to thank Associate Professor Dr. Dev-Prasad Jaisi, sir, for the wonderful review on the understanding of sediment provenance and diagenesis using stable isotope in phosphates. And we are also really pleased to how you have been motivating and how you have been promoting the young generations, and also, yes, your willingness to support the young researchers. We are really happy to hear that, and I believe our viewers, our interested graduates will also take this as a golden opportunity. And we also had a very interesting Q&A sessions with number of quality questions. We tried our best to address all of these questions from our Facebook page live streaming and as well as from our Zoom chat box. So in case if you have any queries, you can please reach out at info@inhrr.org, or you can also visit our website and find the subsequent details of our presenter. I would also like to convey my big thanks to all of our organizing team, speakers, session moderator, and our distinguished guests and participants for participating in this session. The session was really informative and it was also really interactive. We'll be sharing the recorded session on our YouTube channel, and the links will be forwarded to the registered email. We also have a scheduled webinar, which I would like to share now, and it will be regarding the using convergence research to connect 3D architecture of the Himalayan thrust wedge to the seismic hazard and risks in Western Nepal. So I would like to share the poster for this, and it will be presented by the 5 presenters. I think it's gonna be really interesting. So we'll be having Professor Dr. Michael Murphy, Professor Dr. Michael H. Taylor, Dr. Sin Bemis, Dr. Richard Starren, and Dr. Elina J. Sotley. And we'll be having the webinar on September 13th, that is upcoming Sunday. And we'd also like to request our participant to please fill up the feedback survey form, where we'll be able to, you can also suggest us about some potential future webinar topics, and we'll try to address them as possible. So we'd also like to mention that Institute of Himalayan Risk Reduction will be more than happy to collaborate and organize this kind of relevant webinar in the days to come. So with this, I would like to sign up from our session for today. Thank you once again. Thank you so much, everyone. Stay safe. Namaste.
Thank you. Namaste. Bye-bye.
Thank you so much.
See you.
Everyone have a good evening there.
Yeah. Thank you.
Thank you. Goodbye.
Goodbye.